Okay then, so this exam question looks at an experiment investigating the rate of photosynthesis, so the speed of photosynthesis, how quickly it's happening. Okay, now in this experiment, and it comes up in lots of different ways, we always use pondweed. Okay, now we could test the leaf for starch and that proves that photosynthesis is happening, but we can't use that to tell us how quickly it's happening. So what we use instead is the oxygen that's given off. Okay, that's why we use a pondweed, because as the oxygen is given off, it forms bubbles in the water, and we can either count the bubbles or we can actually measure the volume of gas that is collected at the end of the tube. Okay, so it's to do with speed in terms of photosynthesis happening. Now this question asks us, sorry, I'm just having camera difficulties. This question asks us about different colours of light. So it's asking us to investigate how it's affected by different coloured lights. So that is the thing that I am going to change. Okay, now what we need to do is describe the method that we'd use to what we would measure, okay, and what we would control. So if you're controlling it, these are the things that you need to keep the same. Okay, so any experiment is always following exactly the same formula. It's chemistry or biology. You need to think about what it is you're changing, what it is you need to keep the same, and what you will measure for your result. Okay, now it's a 6-mark question, so I need to make sure that I've included a number of different points. Okay, but having those 3 actually helps me to structure how I'm going to approach my answer. So I've got my diagram here. It's sort of half given me what I'm going to do. Okay, so my first point is set up the equipment as shown. Now if I'm testing different colours of light, I must somehow have a lamp near to my pondweed. So I'm going to set up the equipment as shown with a lamp. Now this is one of the things that I need to control, and I need to keep the distance the same, okay, with the lamp to the beaker. So I'm going to give a distance. So I'm going to say the lamp is at 20 centimetres from the beaker. So I've already listed something that I need to control. Okay, then what I'm going to do is I'm going to measure. So what am I measuring? I can either measure the volume or I can, sorry, let me just turn my phone off. I can either measure the volume or I can count the bubbles. Now counting bubbles is probably the easiest thing, okay, but I need to be specific in terms of how long I'm counting them for. So again, I'm just going to pick a time, right? So I'm going to count the number of bubbles in 2 minutes. Now you could say 1 minute, you could say 5 minutes. It's entirely up to you, okay? But I've said something. So this is something I'm keeping the same. This is something I'm keeping the same. And then I'm measuring my number of bubbles. So I'm doing it for 2 minutes, okay? Now, so that I can have valid results that are reliable, that I can trust them, particularly in biology, we need to repeat it, okay? So I'm going to repeat this experiment 3 times and calculate the average number, oh I missed my A out of average there, number of bubbles. Okay, so I've got a much more reliable result if I can work out an average. Now I need to go back to the thing that I'm going to change. Okay, so I've kept some things the same, I've measured something, but I haven't changed anything yet. So I need to change the different colour of light. Okay, so what my last point would be is that I'm going to repeat the experiment with different coloured bulbs or filters that you can put in front of the lamp so that you can get the different coloured light on your pondweed. Okay, so now I've written 4 points, but I've included all the parts that they've asked me to do. So I've said what I'm going to change, I've said what I'm going to measure, that's the number of bubbles, and I've said some things that I'm going to keep the same. Okay. Now this experiment can also come up as investigating light intensity, and what you would do is exactly the same, but instead of changing the colour of the light, what you would do is that you would have your lamp, sorry, this is a really badly drawn lamp, you'd have your lamp next to it, and what you would do is you'd move it further away. Okay, so that's an alternative way of doing this experiment, but you are very likely to see something like this, okay, and be asked to write a method, right, but as long as you follow exactly the same formula. So what is it you're changing, what is it you're measuring, and what is it you're keeping the same, okay, then you will get a good number of marks. Okay, so that's how we write methods for experiments.